Welcome back to the deep dive. Today, we're diving into something, well, it's a bit of a technical topic, but super important,
Yeah,
especially if you're into cannabis or psychedelics.
definitely.
It's analytical method development and validation.
It might sound a little dry, but trust me, it really matters. It affects what's actually in that gummy or that microdose you're taking.
Okay, so we're talking about like making sure what's on the label is actually in the product.
Exactly.
Like how much THC in a gummy or psilocybin in a capsule.
Yeah, you got it. It's all about accuracy, reliability, and safety.
So it's not as simple as just like throwing it on a scale.
No, no, not even close. Especially with complex mixtures like cannabis extracts or psilocybin mushrooms.
Right.
Remember all those different compounds we talked about in previous deep dives?
Oh yeah, the whole molecular machinery thing. It's not just THC or psilocybin. There's a whole bunch of other stuff in there.
Yeah, exactly. And that's where analytical method development comes in. It's about creating specific, accurate, and reliable ways to measure not only the main active ingredient, but also all those other compounds and any potential impurities.
So it's like creating a detailed recipe for how to analyze these substances.
Yeah, you could say that.
And then validation is making sure that recipe actually works.
Exactly. And those recipes have to meet strict guidelines.
Okay.
That's where the ICHQ2 guideline comes in. It's like the gold standard for validating these methods.
ICH. What does that stand for again?
The International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use.
Catchy.
It's a mouthful, but basically they set the standards for pharmaceutical testing globally. And these standards are being applied more and more to cannabis and psychedelics as they become more widely used, especially for medical purposes.
So this ICHQ2 guideline, it's like a checklist to make sure your testing methods are good.
Yeah, like a quality control checklist.
What kind of things are on that checklist?
Well, there are 6 key parameters: specificity, accuracy, precision, linearity, range, and robustness.
6 parameters, okay.
They all play a crucial role in ensuring reliable and accurate test results, which are critical for safety and efficacy.
Okay, let's break those down one by one. What's specificity mean in this context?
Specificity means your testing method can target precisely what you're looking for, even in a complex mixture, like picking out a specific face in a crowded room.
Okay.
If you're testing cannabis extract, a specific method would be able to distinguish THC from CBD, making sure you're measuring the right thing.
So no accidental mix-ups.
Yeah.
Got it. What about accuracy? That's just making sure your measurement is correct.
Yeah, spot on. Accuracy is how close your measurement is to the true value. Think of it like hitting the bullseye on a target.
Makes sense.
You want your method to be as accurate as possible for reliable results.
Okay, and then precision is about consistency, like
Yeah.
hitting the same spot on the target repeatedly, even if it's not the bullseye.
Perfect analogy. Precision is all about reproducibility, getting the same measurement every time. It's crucial because if your method isn't precise, you can't be confident in the data.
Okay, so we've got specificity, accuracy, and precision. What's next on this ICHQ2 checklist?
Linearity. This means your method can provide accurate results across a range of concentrations. Think about testing different cannabis products with different potencies, from a low-dose CBD oil to a high THC concentrate.
Right.
A linear method would give you accurate results for all of them.
So no matter how strong or weak the sample is, the method should still work accurately, like a ruler that works for inches and miles.
Exactly. And related to linearity is range.
Okay.
This refers to the span of concentrations over which your method is valid, the low and upper limits of your testing sweet spot, the concentrations where your method consistently produces reliable results.
So if you're testing a microdose of psilocybin, your method needs to be sensitive enough to accurately measure those tiny amounts. And if you're testing a high-potency cannabis extract, it needs to be able to handle those higher concentrations without getting thrown off.
Exactly. Defining the range is essential to making sure your method is fit for purpose.
Makes sense. We've covered 5 of the 6 parameters. What's the final one on this ICHQ2 checklist?
Robustness. This is all about how well your method can handle real-world variability. Labs aren't perfect environments. There might be slight temperature changes, slight variations in equipment, or even differences in how technicians perform the tests.
So robustness is like a stress test for your method.
Yeah, you could say that.
Seeing how well it holds up when things aren't perfect?
Yeah,
If it's robust, you can be confident those little variations won't mess up your results.
exactly. This is particularly important for cannabis and psychedelics because they're complex and variable in their composition.
Okay.
A robust method ensures you're getting consistent, reliable results, even with real-world variations.
Okay, I'm starting to see why this stuff is so important. It's like building a house. You need a solid foundation and reliable materials to make sure it's safe and sturdy.
Yeah.
And in this case, ICHQ2 provides the blueprint for that foundation,
I like that analogy.
making sure the methods used to test these substances are accurate and trustworthy.
It all ties back to protecting consumers
Right.
and ensuring they have access to safe and effective products. Remember those discussions about labeling errors and insufficient testing? That's where things can get tricky.
Oh yeah, those examples were a bit concerning.
Definitely.
It really highlighted why this topic matters in the real world.
Absolutely. One of the key takeaways from those sources was that if testing methods aren't up to par, you get labeling errors,
Right.
where the actual potency or purity of a product doesn't match what's on the label.
And that can be a problem for a lot of reasons.
Yeah, for sure.
Someone might not be getting the dose they think they are, or they could be unknowingly exposed to harmful impurities.
Exactly. That's why robust analytical method development and validation are essential. It's about ensuring consumer safety, building trust in the industry, and promoting responsible use of these substances.
So we've covered the basics of why this is important and the ICH guidelines.
Right.
But I'm still wondering, how does all this actually play out in practice? What are some of the specific challenges of analyzing these complex mixtures?
Right.
And what kind of technology is involved?
Yeah, it's one thing to talk about guidelines and parameters, but I imagine it's a whole other thing to actually put them into practice.
Oh, absolutely.
Especially with something as complex as cannabis or psilocybin.
Yeah, analyzing these substances, it's not like baking a cake,
Right.
We're not just measuring one or two ingredients.
Right.
We're dealing with a whole symphony of compounds.
It's like that molecular machinery we talked about.
Exactly.
But now we're trying to take it apart and figure out how each piece works
Yeah.
and how they all interact with each other.
Precisely. And to do that, we need some pretty sophisticated tools and techniques.
One of the most common is high-performance liquid chromatography, or HPLC.
HPLC. That rings a bell from chemistry class. What does it actually do?
Imagine you have a mixture of different compounds dissolved in a liquid.
Yeah.
HPLC is like a super efficient sorting system. It separates those compounds based on their chemical properties. Kind of like a microscopic racetrack where each compound moves at a slightly different speed.
Okay.
And that allows us to identify and quantify them individually.
So you can see exactly what's in the mix.
Yes.
And how much of each compound is there.
Precisely. And often, HPLC is paired with another technique called mass spectrometry, or MS.
Okay.
This adds another layer of identification by measuring the mass-to-charge ratio of each compound. It gives it a unique fingerprint that helps confirm its identity.
So HPLC sorts them out, and then MS gives each one like a unique ID tag.
Yeah, exactly.
They're like the dynamic duo of chemical analysis.
That's a great way to put it.
And this level of analysis is essential for understanding the full complexity of these substances, right?
Absolutely.
It's not just about measuring the main active ingredient, but getting a complete picture of everything that's in there,
It's about the whole profile.
including those other compounds that might contribute to the overall effects.
Exactly.
And of course, identifying any potential impurities. We talked about those earlier.
Yes.
I'm guessing they can be particularly tricky to detect.
You're right. Impurities can be sneaky. Some can be more concerning than others. For example, mutagenic impurities, which can potentially cause changes in DNA, are a major focus.
Mutagenic impurities, that sounds pretty serious.
Yeah.
How do those even get into the products?
Well, they can come up during the manufacturing process
Oh,
from
right.
the raw materials or the chemical reactions involved in making or extracting the substances.
Right.
It's complicated, but the bottom line is that controlling them is essential for safety.
So not just getting like a bad batch of gummies, but potentially dealing with long-term health risks.
Exactly.
That really highlights why this testing is so important.
It is. And what's interesting is researchers are constantly developing new and more sensitive techniques to detect these impurities at really low levels.
Wow.
They're exploring things like nuclear magnetic resonance, spectroscopy, or NMR.
NMR.
Yeah.
That's the one with the giant magnets, right?
Yes.
Yeah.
It uses powerful magnets to analyze the structure and properties of molecules.
Cool.
And when it comes to impurities, this level of sensitivity is crucial.
Yeah.
Especially as cannabis and psychedelics move closer to mainstream medical use.
Right.
We need to be absolutely sure that they're as safe as possible.
Okay, so we've delved into the complexities of analyzing these mixtures and the tech involved.
Yeah.
But how does this translate to the real world? Can you give some examples of what can go wrong when testing isn't good enough?
Absolutely. Remember those labeling errors we mentioned earlier? There have been cases where cannabis products, for instance, were labeled as having a specific concentration of THC, but independent testing found that the actual potency was way off.
So, like, the label says one thing, but what's actually in the product is totally different.
Yeah, exactly.
Unsettling.
It is.
And it highlights the importance of accurate testing throughout the whole supply chain.
For sure.
If the labeling is wrong, it can have real consequences for consumers,
Yeah,
especially those using these products for medical reasons.
definitely.
You imagine someone relying on a specific dose of CBD for anxiety,
Right.
but the product they're using isn't what they think it is.
Yeah,
That could really impact their well-being.
absolutely. Or someone thinking they're taking a low dose when it's actually much stronger.
Right.
That could be really disorienting or even dangerous, especially for someone new to these substances
Yeah.
or someone with a specific medical condition.
And it's not just about the potency of the main active ingredient.
Right.
Insufficient testing could also mean missing harmful impurities.
Exactly.
So it's really about transparency and accountability.
Absolutely.
Consumers need to be able to trust that what's on the label is accurate.
Yes.
And that it's been thoroughly tested
For sure.
for both potency and purity.
Absolutely. And as the industry grows and evolves, this focus on quality control and accurate testing will only become more important.
Right.
It's the foundation for building trust, ensuring consumer safety, and unlocking the full potential of these substances.
Okay, so we've covered a lot of ground here.
Yeah.
We've talked about the importance of analytical method development and validation,
Right.
those ICH guidelines, the challenges of analyzing complex mixtures, and the potential consequences of insufficient testing.
And all of this leads us to a really interesting question.
Okay?
What does the future hold for this field?
Yeah, that is a great question.
As cannabis and psychedelics become more mainstream, what new challenges and innovations can we expect to see?
Yeah, it feels like we're at a turning point.
It does.
I'm curious to know what's next.
Me too.
What do you think? Any exciting developments in the pipeline? I'm ready for those glimpses into the future. What's on the horizon for analytical testing?
One area I'm really excited about is personalized medicine.
Personalized medicine, like tailoring treatments to each person? I thought that was mainly for traditional medicine.
It has been, but we're starting to see that applied to cannabis and psychedelics, too. Imagine analyzing not just the product's chemistry, but also a person's genes and how they might metabolize these substances.
Wow, that's next level. So instead of a one-size-fits-all, you could have personalized doses,
Yeah,
specific products tailored to a person's needs,
exactly.
and even predict how someone might respond to different substances.
Exactly. It's about taking those ICH guidelines and making them personal.
Okay.
Not just making sure the product is safe and labeled right, but tailoring the whole experience to a person's biology.
Okay, I'm on board with this future.
Right.
But how would that even work? How could you personalize something like cannabis therapy?
It comes down to combining chemical analysis with genetic testing.
Okay.
So you'd know the exact composition of the product and how someone's genes and metabolism might influence the effects. For example, some people might process THC faster than others, meaning they might need a higher dose to feel the same effects.
So it's like taking into account all that molecular machinery,
Yeah,
but on a personal level. That's amazing.
it really is.
And it opens up so many possibilities for optimizing well-being.
Absolutely.
It's using these substances to improve people's lives responsibly and in a really personalized way.
Exactly.
Okay, I'm sold. But let's be real, that's huge. It would need experts from all sorts of fields working together.
It is multidisciplinary.
Chemists, geneticists, doctors, the whole works.
So you need
Everything it's actually possible.
The research is moving so fast, and with legalization growing, I think we have a real chance to unlock this personalized potential.
It's amazing to think that what seemed like a niche area of research,
Right.
analytical method development, could actually be leading this revolution in medicine and well-being.
Yeah.
All because of those guidelines and methods, making sure everything's accurate and safe.
It all comes back to that foundation, that
Right.
commitment to quality and safety.
Yeah.
Those numbers on a label, the data from the instruments they represent, making sure these substances are used responsibly.
Well, there you have it, folks.
Yep.
We've taken a deep dive into analytical method development and validation.
We have.
And I think it's safe to say it's way more exciting than it sounds at first.
I agree. Much more interesting than it might seem.
We talked about why accurate testing is so important,
Yes.
how to analyze these complex mixtures,
Yeah.
and even peeked into the future of personalized medicine.
The future is bright,
It is.
and science is leading the way.
So the next time you see a cannabis product or think about trying psilocybin, remember those ICH guidelines?
Yeah.
And the science behind making sure what you're getting is safe and actually what it says on the label.
And as this field keeps evolving, stay curious, stay informed, and keep an open mind about the potential these substances have.
Thanks for joining us on this deep dive. Until next time, keep exploring, keep learning, and keep those mind buzzing.